Chapter 4 Psychosis and Schizophrenia. Let's start with symptom dimensions in schizophrenia and clinical description of psychosis. Psychosis is a syndrome, that is, it is a mixture of symptoms. It can be associated with many different psychiatric disorders, but is not a specific disorder in itself in any diagnostic schemes such as DSM-5 or ICD-10. At the very basic, psychosis means delusions and hallucinations, and generally also includes symptoms such as disorganized speech, disorganized behavior, and gross distortion of reality. So on the whole, psychosis can be considered to be a set of symptoms in which a person's mental capacity, affective response, capacity to recognize reality, to communicate, and to relate to others is impaired. The disorders that require the presence of psychosis as a defining feature of the diagnosis include schizophrenia, substance-induced psychotic disorders, schizophreniform disorder, schizoaffective disorder, delusional disorder, brief psychotic disorder, and psychotic disorders due to a general medical condition. Disorders that may or may not have psychotic symptoms as an associated feature include mania, depression, as well as several cognitive disorders such as Alzheimer's dementia. Now, psychosis itself can be paranoid, disorganized, or depressive. And perceptual distortions and motor disturbances can be associated with any type of psychosis. Now, perceptual distortions include being distressed by, first, hallucinatory voices, that is, hearing voices that accuse, blame, or threaten punishment, or seeing visions, having hallucinations of touch, taste, or odor, or seeing that familiar things and people seem changed. Motor disturbances include peculiar rigid postures, overt signs of tension, inappropriate grins or giggles, peculiar repetitive gestures, talking, muttering, or mumbling to oneself, or glancing around as if hearing voices. Now let's discuss paranoid psychosis, in which a patient might have paranoid projections, hostile belligerence, and grandiose expansiveness. In paranoid projections, the patient might be preoccupied with delusional beliefs, believing that people are talking about oneself, or believing one is being persecuted or being conspired against, or that people or external forces control one's actions. Now, hostile belligerence might be expressed in the following ways: feeling or manifesting hostility, attitude of disdain, sullen attitude, manifesting irritability or grouchiness, tending to blame others for problems, expressing feelings of resentment, or finding fault and expressing suspicion of people. And finally, grandiose expansiveness is exhibited by attitude of superiority, hearing voices that praise and extol, believing one has unusual powers, or is a well-known personality, or one has a divine mission. Now let's discuss the second type of psychosis, which is disorganized or excited. In this, there is conceptual disorganization, disorientation, and excitement. Conceptual disorganization can be characterized by giving answers that are irrelevant or incoherent, drifting off the subject, using neologisms, or repeating certain words or phrases. And disorientation is usually tested on the basis of time, place, and person, as in, the patient might not be knowing where one is, what is the season of the year, or which year it is, or even one's own age. Now, in excitement, the patient might be expressing feelings without restraint, may manifest speech that is hurried, may exhibit an elevated mood, may have an attitude of superiority, dramatizing oneself or one's symptoms, may manifest loud and boisterous speech, may exhibit overactivity or restlessness. Now let's discuss the third type of psychosis, which is depressive, which is characterized by psychomotor retardation and apathy, and second, anxious self-punishment and blame. Now, psychomotor retardation and apathy are manifested by the following: slowed speech, indifference to one's future, fixed facial expression, slowed movements, deficiencies in recent memory, blocking in speech, slovenly appearance, whispered speech, or even failure to answer questions. Now, anxious self-punishment and blame is the tendency to blame or condemn oneself, anxiety about specific matters, apprehensiveness regarding vague future events, an attitude of self-deprecation, manifesting as a depressed mood, expressing feelings of guilt or remorse, preoccupation with suicidal thoughts, unwanted ideas, and specific fears, and even feelings of being unworthy and sinful. Schizophrenia is more than psychosis. It affects around 1% of the population. Between 25 to 50% of schizophrenia patients attempt suicide, and 10% eventually succeed, contributing to a mortality rate of 8 times greater than that of the general population. Now, life expectancy of a patient with schizophrenia may be 20 to 30 years shorter than that of general population, not only due to suicide but in particular due to premature cardiovascular disease caused not only by genetic and lifestyle factors such as smoking, unhealthy diet, and lack of exercise leading to obesity and diabetes, but also from treatment with some antipsychotic drugs which themselves cause an increased incidence of obesity and diabetes and thus increase cardiac risk. Schizophrenia by definition is a disturbance that must last for 6 months or longer, including at least 1 month of delusions, hallucinations, disorganized speech, grossly disorganized or catatonic behavior, or negative symptoms. Now coming on to positive symptoms of psychosis and schizophrenia. These include delusions, hallucinations, distortions or exaggerations in language and communication, disorganized speech, disorganized behavior, catatonic behavior, or agitation. Now these can erupt suddenly when a patient decompensates into a psychotic episode, often called a psychotic break, as in break from reality. Now coming on to delusions, these are a type of positive symptoms and usually involve misinterpretation of perception or experiences. The most common content of delusions in schizophrenia are persecutory, self-referential themes, somatic, religious, or grandiose. Now hallucinations may occur in any sensory modality, that is auditory, visual, olfactory, gustatory, or even tactile. But auditory hallucinations are by far the most common and characteristic. Now distortions and exaggerations in language and communication, that is disorganized speech, as well as in behavior, that is grossly disorganized, catatonic, or agitative behavior may also occur. Now coming on to negative symptoms. The first one is alogia, that is poverty of speech. That means talks little or uses few words. Now the second one is affective blunting, that is reduced range of emotions in perception, experience, or expression. For example, the patient might say that he feels numb or empty inside, may recall very few emotional experiences, good or bad. The third one is asociality, that is reduced social drive and interests. In this, patient has little sexual interest, has few friends, and shows little interest in spending time with them. The fourth one is anhedonia, that is reduced ability to experience pleasure. In this, the patient finds previous hobbies or interests as unpleasurable. The fifth is avolition, that is reduced desire, motivation, or persistence. In this, the patient has reduced ability to undertake and complete everyday tasks and even may have poor personal hygiene. Although this reduction in normal functioning may not be as dramatic as positive symptoms, it is interesting to note that negative symptoms determine whether a patient ultimately functions well or has a poor outcome. And thus, negative symptoms determine whether a patient with schizophrenia can live independently, maintain a stable social relationship, or re-enter the workplace. Now, negative symptoms are not just part of the syndrome of schizophrenia, but they can also be a part of the prodrome that begins with subsyndromal symptoms that do not meet the diagnostic criteria of schizophrenia and occur between the onset of the full disease. And thus, these are very important to detect and monitor over time in high-risk patients so that treatment can be initiated at the first sign of psychosis. And finally, the negative symptoms can also persist between psychotic episodes. Now let's discuss key negative symptoms that can be identified solely on observation. The first one is reduced speech. In this, the patient has restricted speech quantity, uses few words and non-verbal responses, has impoverished content of speech where words convey little meaning. The second one is poor grooming. In this, the patient has poor hygiene, his clothes are dirty or stained, or the patient actually has an odor. The third one is limited eye contact, in which the patient rarely makes eye contact with the interviewer. Now coming on to key negative symptoms that can be identified with some questioning. The first one is reduced emotional responsiveness. In this, the patient exhibits few emotions or change in facial expression when questioned and can recall few occasions of emotional experience. The second one is reduced interest. In this, the patient has reduced interests and hobbies. There is little or nothing that stimulates his interest, and he has limited life goals or an inability to proceed with them. The third one is reduced social drive. In this, the patient has reduced desire to initiate social contacts and may have few or no friends or close relationships. Now, instituting treatment for negative symptoms during the prodromal phase of schizophrenia may delay or prevent the onset of the illness, but is still a matter of current research. Beyond positive and negative symptoms of schizophrenia. Although not recognized formally as part of the diagnostic criteria for schizophrenia, numerous studies subcategorizes the symptoms of this illness into 5 dimensions, not just 2. And the other 3 are the cognitive symptoms, aggressive symptoms, and affective symptoms. Now let us discuss the first, aggressive symptoms. Now these include assaultiveness, verbal abusive behavior, and frank violence. For this, behavioral interventions may be particularly helpful to prevent violence linked to impulsivity by reducing provocations from the environment, as well as certain antipsychotic drugs such as clozapine or very high doses of standard antipsychotic drugs, or occasionally the use of 2 antipsychotic drugs simultaneously may also be helpful in aggressive symptoms. Now coming on to cognitive symptoms of schizophrenia. Now these are impaired attention and information processing that is manifested as impaired verbal fluency, problems with serial learning, impairment in vigilance for executive functioning, that is, problems with sustaining, focusing, concentrating, and prioritizing attention, and finally, inability to modulate behavior based upon social cues. The second domain is executive dysfunction, which includes problems with representing and maintaining goals, allocating attention resources, evaluating and monitoring performances, and utilizing these skills to solve problems. Now, these cognitive symptoms of schizophrenia are important to recognize and monitor because they are the single strongest correlate of real world functioning, even stronger than negative symptoms. Now let's once again summarize the cognitive symptoms of schizophrenia. These are problems representing and maintaining goals, problems allocating attention resources such as focusing, sustaining, dividing and shifting attention, problems evaluating, monitoring and prioritizing functions and performances, impaired verbal fluency, problems modulating behavior based upon social cues, difficulty with serial learning, and finally, difficulty with problem solving. Now coming on to the affective symptoms of schizophrenia. These are similar to symptoms associated with either mania, depression, or both. Now different symptom domains of schizophrenia are hypothesized to be regulated by unique brain regions. The positive symptoms of schizophrenia are hypothetically modulated by malfunctioning of mesolimbic circuits, and the negative symptoms are hypothetically linked to malfunctioning of mesocortical circuits and may also involve some mesolimbic regions, such as nucleus accumbens, which is part of the brain's reward circuit and thus plays a role in motivation. It may also be involved in the increased rate of substance use and abuse seen in patients with schizophrenia. Affective symptoms are said to be associated with the ventromedial prefrontal cortex. The aggressive symptoms related to impulse control are associated with abnormal information processing in the orbitofrontal cortex and amygdala. And finally, the cognitive symptoms are said to be associated with problematic information processing in the dorsolateral prefrontal cortex. Although there is overlap in function among different brain regions, understanding which brain regions may be predominantly involved in specific symptoms can aid in customization of treatment. to the particular symptom profile of each individual patient. Symptoms of schizophrenia are not unique to schizophrenia. Positive symptoms can also occur in bipolar disorder, schizoaffective disorder, psychotic depression, Alzheimer's disease, other organic dementias, childhood psychotic illnesses, and drug-induced psychosis, and others. Negative symptoms can also occur and overlap with cognitive and affective symptoms in autism, post-stroke dementia, Alzheimer's disease, and many other organic dementias. And as we know, affective symptoms are prominent features of major depressive disorder, psychotic depression in bipolar disorder, schizoaffective disorder, organic dementias, and childhood psychotic disorders. And aggressive and hostile symptoms are frequently associated with bipolar disorders, childhood psychosis, borderline personality disorder, antisocial personality disorder, drug abuse, Alzheimer's and other dementias, attention deficit hyperactivity disorder, and conduct disorder in children. Now let's discuss brain circuits and symptom dimension in schizophrenia. The positive symptoms of schizophrenia have long been hypothesized to be localized to malfunctioning of mesolimbic circuits, especially involving the nucleus accumbens. The nucleus accumbens is considered to be part of the brain's reward circuit, so it is not surprising that problems with reward and motivation in schizophrenia can lead to smoking, drug, and alcohol abuse. The prefrontal cortex is considered to be a key node in the nexus of malfunctioning cerebral circuitry responsible for each of the remaining symptoms in schizophrenia. Specifically, the mesocortical and ventromedial prefrontal cortex with the negative symptoms and affective symptoms, the dorsal prefrontal cortex with cognitive symptoms, and the orbital frontal cortex and its connections to amygdala with the aggressive and impulsive symptoms. Now this model is obviously oversimplified and reductionist, but has both heuristic and clinical value, useful to optimize and individualize treatment. It can be very useful to consider which specific symptoms any given patient is expressing, and therefore which areas of that particular patient's brain are hypothetically malfunctioning. And it can also assist the clinician in choosing medication and monitoring the effectiveness of the treatment. Now let's discuss neurotransmitters and circuits in schizophrenia. Dopamine is synthesized in dopaminergic nerve terminals from amino acid tyrosine after it is taken up into the neurons from the extracellular spaces and bloodstream by tyrosine pump or transporter. Tyrosine is first converted into DOPA by the rate-limiting enzyme tyrosine hydroxylase, which is then converted into dopamine by enzyme DOPA decarboxylase, as shown in this flowchart. The dopamine thus generated is then taken up into synaptic vesicles by vesicular monoamine transporter, also known as VMAT2, and stored there until it is used during neurotransmission. The dopamine neurons also has a presynaptic transporter, also known as reuptake pump, called DAT, which is unique to dopamine and which terminates dopamine's synaptic action by whisking it away from the synapse. Now, a point of interest is that in prefrontal cortex, DATs are relatively sparse and dopamine is inactivated by other mechanisms. So, the excess dopamine that escapes storage in synaptic vesicles can be destroyed within the neuron by enzyme monoamine oxidase A or B that is present in the mitochondria within the presynaptic neurons, and outside the neuron by enzyme catechol O-methyltransferase, also known as COMT. Now interestingly, dopamine that diffuses away from the synapse can also be transported by norepinephrine transporters, that is NETs, as a false substrate. The DAT and the vesicular transporters VMAT2 are both type of receptors, and at least 5 pharmacological subtypes of dopamine receptors exist. The most extensively investigated dopamine receptor is the dopamine 2 or D2 receptor, as it is stimulated by dopamine agonist, as in for the treatment of Parkinson's disease, and blocked by dopamine antagonists, as in antipsychotics for the treatment of schizophrenia. Now D2 receptors can be presynaptic also, where they function as autoreceptors and acts as gatekeepers, either allowing DA release when they are not occupied by DA or inhibiting DA release when DA builds up in the synapse and occupies these gatekeeping presynaptic autoreceptors. Now, such receptors are located, as we know, either on the axon terminal or on the other end of the neuron, as in the somatodendritic area. Now, as just mentioned, these D2 autoreceptor can also be located in somatodendritic areas. When dopamine binds to these autoreceptors, it shuts off neuronal impulse flow in the dopamine neuron. Thus, in both cases, occupancy of these D2 receptors provide negative feedback, input or braking action. Now, let's discuss some key dopamine pathways in the brain. There are 5 main dopamine pathways. These are mesolimbic, mesocortical, nigrostriatal, tuberoinfundibular, and a 5th pathway that innervates the thalamus. Now let's discuss dopamine hypothesis of schizophrenia and mesolimbic dopamine pathway and positive symptoms. The mesolimbic dopamine pathway projects from the dopaminergic cell bodies in the ventral tegmental area of the brainstem to the axon terminals in one of the limbic regions of the brain, namely nucleus accumbens in the ventral striatum, as shown here. Now, this pathway has an important role in several emotional behaviors, including the positive symptoms of psychosis, such as delusions and hallucinations. Also, it is very important for motivation, pleasure, and reward. Now, it has been observed that diseases or drugs that increase dopamine will enhance or produce positive psychotic symptoms, whereas drugs that decrease dopamine will reduce or stop positive symptoms. It is very interesting that stimulant drugs such as amphetamines and cocaine release dopamine and, if given repetitively, can cause paranoid psychosis, which is virtually indistinguishable from positive symptoms of schizophrenia. All known antipsychotic drugs capable of treating positive psychotic symptoms are blockers of dopamine D2 receptors. Thus, hyperactivity of the mesolimbic dopamine pathway hypothetically accounts for positive symptoms, whether those symptoms are part of the illness of schizophrenia, drug-induced psychosis, mania, depression, or dementia. Furthermore, hyperactivity of mesolimbic dopamine neurons may also play a role in aggressive and hostile symptoms, especially if the serotonergic control of dopamine is aberrant in patients who lack impulse control. Current theories now state that it is the downstream consequence of dysfunction in prefrontal cortex and hippocampal glutamate activity. Now, coming on to the mesocortical dopamine pathway and the negative, cognitive, and affective symptoms of schizophrenia. Now, this pathway also arises from the cell bodies in the ventral tegmental area but project to the prefrontal cortex, and of course it is known as the mesocortical dopamine pathway. Branches of this pathway into the DLPFC are hypothesized to regulate cognitive and executive functions, whereas the branches of this pathway to the VMPFC are hypothesized to regulate emotion and affect. To reiterate, the cognitive and some negative symptoms of schizophrenia may be due to a deficiency of dopamine activity in the mesocortical projections to the DLPFC area of the cortex, whereas the affective and other negative symptoms of schizophrenia may be due to a deficit of dopamine activity in mesocortical projections to VMPFC. Now, lack of this proper functioning of mesocortical dopamine projections may be the consequence of neurodevelopmental abnormalities in the NMDA glutamate system. Now, let's discuss the mesocortical dopamine hypothesis of negative, cognitive, and affective symptoms of schizophrenia. Now, the two pathways we just discussed poses a therapeutic dilemma: how to increase dopamine in the mesocortical pathway while simultaneously decreasing dopamine activity in the mesolimbic dopamine pathway. Now, let's discuss mesolimbic dopamine pathway, reward, and negative symptoms. When a patient with schizophrenia loses motivation and interest and has anhedonia, also implicates a deficient functioning of the mesolimbic dopamine pathway. Now, interestingly, the prefrontal cortex area does not have a high density of D2 receptors. This may cause inadequate reward mechanism, which can be seen as behaviors such as anhedonia, drug abuse, and negative symptoms which is exhibited as lack of rewarding social interactions and lack of general motivation and interest. Perhaps the much higher incidence of substance abuse in schizophrenia than in healthy adults, especially of nicotine, but also of stimulants, could be partially explained as an attempt to boost the functioning of defective mesolimbic dopaminergic pleasure centers. Now coming on to the third pathway, that is the nigrostriatal dopamine pathway. Now this pathway projects from the dopaminergic cell bodies in substantia nigra, that is in the brainstem, via axons terminating in basal ganglia in the striatum. As is well known, this is a part of extrapyramidal nervous system and controls motor movements. As can be expected, deficiency of dopamine in this pathway causes movement disorders, including Parkinson's disease, which is characterized by rigidity, akinesia or bradykinesia, and tremors. Now, dopamine deficiency in the basal ganglia can also produce akathisia, which is a type of restlessness, and dystonia, that is twisting movements, especially of the face and neck. These movement disorders can be replicated by drugs that block D2 receptors in this pathway. And on the opposite end, the hyperactivity of dopamine in the nigrostriatal pathway is thought to underlie various hyperkinetic movement disorders such as chorea, dyskinesia, and tics. And chronic blockade of D2 receptors in this pathway may result in a hyperkinetic movement disorder known as neuroleptic-induced tardive dyskinesia. Now let's discuss the fourth pathway, that is the tuberoinfundibular dopamine pathway. This pathway projects from hypothalamus to anterior pituitary, and normally these neurons are active and inhibit prolactin release. But in postpartum state, however, the activity of these dopamine neurons is decreased. Prolactin levels can therefore increase and breastfeeding and lactation can occur. And thus, if the functioning of these neurons is disrupted by lesion or drugs, can lead to increased prolactin levels. And elevated prolactin levels are associated with galactorrhea, amenorrhea, and sexual dysfunction. And understandably, these problems can occur after treatment with many antipsychotic drugs. And thus oddly, in untreated schizophrenia, the functioning of this tuberoinfundibular pathway may be relatively preserved. Now coming on to the final, thalamic dopamine pathway. Now this pathway arises from multiple sites, including the periaqueductal gray matter, the ventral mesencephalon, various hypothalamic nuclei, and lateral parabranchial nucleus. Its function is still under investigation, but it may be involved in sleep and arousal mechanisms by gating information passing through the thalamus to cortex and other brain areas. Now let's discuss glutamate. Understandably, glutamate has attained a key theoretical role in the hypothesized pathophysiology of schizophrenia, as well as in a number of other psychiatric disorders, including depression. Now is also a key target to novel psychopharmacological drugs. Now glutamate is the major excitatory neurotransmitter in the central nervous system and sometimes considered to be the master switch in the brain, since it can excite and turn on virtually all CNS neurons. Let's discuss glutamate synthesis. Now glutamate or glutamic acid is a neurotransmitter which is actually an amino acid. It is synthesized from glutamine in glia cells, which also assist in the recycling and regeneration of more glutamate. After it interacts with receptors in the synapse, it is taken up into the neighboring glia cells by a reuptake pump known as the excitatory amino acid transporter or EAT. After reuptake into glia, the glutamate is converted into glutamine by the enzyme known as glutamine synthetase. And the same process can be done by another transporter known as the alanine serine cysteine transporter or ASCT. Now this glutamine can again be released from the glia cells by a pump or transporter known as specific neutral amino acid transporter or SNAT. Therefore, when the SNATs and ASCTs operate in inward direction, they transport glutamine and other amino acids into glial or other neurons. But interestingly, they can also be reversed so that the glutamine can get out of the glia and hop a ride into a neuron via a different type of neuronal SNAT operating inwardly in a reuptake pump manner. And once inside the neuron, the glutamine is converted back into glutamate for use as neurotransmitter by an enzyme in mitochondria called the glutaminase, as is shown in the following diagram. The glutamate is then transported into synaptic vesicles via the V-glutes where it is stored. Once released, the glutamate's actions are stopped not by enzymatic breakdown, as is the case with other neurotransmitter systems, but by removal by IATs on neurons or glia. Now let's discuss synthesis of glutamate co-transmitters glycine and D-serine. Glutamate systems are curious in that one of the key receptors for glutamate requires co-transmitters in addition to glutamate in order to function. That receptor is the NMDA receptor, and the co-transmitters is either the amino acid glycine or D-serine. The glutamate neurons get the glycine they need for the NMDA receptors either from the glycine neurons or from the glia. Now the glycine is unable to diffuse very far from the neighboring glycine neurons, as they are taken back into those neurons by a type of glycine reuptake pump known as the type 2 glycine transporter or GLIT2. Thus, the neighboring glia is thought to be the source of most of the glycine available for glutamate synapses. Now this glycine can be taken up into the glia as well as the glutamate neurons from the synapse by type 1 glycine transporter or GLIT1. It can also be taken up into the glia by glial SNAT. Interestingly, it is not known to be stored within synaptic vesicles of glia. But this glycine in the cytoplasm of glia is nevertheless somehow available for release into the synapse, and it escapes these glial cells by riding the aforementioned GLIT1 transporter. Thus, the GLIT1 transporter transport glycine both in and out of glial cells. Once outside, the glycine can get right back into the glia by an inwardly directed GLIT1 and is thought to be the main mechanism responsible for terminating the action of synaptic glycine. But in contrast, D-serine is thought to be stored within some type of synaptic vesicle within the glia. There are novel treatments for schizophrenia that boost glycine action and thus glutamate action at NMDA receptors. And these include inhibitors of key glycine transporter GLIT1 and selective glycine reuptake inhibitors or SGRIs. Glycine can also be synthesized from the amino acid L-serine, transported into the glial cells by an L-serine transporter known as L-sert, and converted from L-serine into glycine by a glial enzyme known as serine hydroxymethylase transferase or SHMT. As can be seen by the equation, this enzyme also works in both directions, either converting L-serine into glycine or glycine into L-serine. Now D-serine is very unusual as the 20 known essential amino acids are all L-amino acids. It just so happens that D-serine has high affinity for the glycine site on NMDA receptors, and that glia are equipped with an enzyme called D-serine racemase that converts the regular L-serine into the neurotransmitter amino acid D-serine and vice versa. Thus, D-serine can be derived either from glycine or from L-serine. Interestingly, the D-serines are produced may be stored in some sort of vesicle in the glia for subsequent release on a reversed glial D-serine transporter or D-sert. Now D-serine's actions are not only terminated by the synaptic reuptake where the inwardly acting glial D-sert, but also by the enzyme D-amino acid oxidase or DOA that converts D-serine into the inactivated hydroxypyruvate. Now brain makes an activator for DAO known not surprisingly as D-amino acid oxidase activator or DAOA. Gene that makes DAOA may be one of the most important regulatory genes that contribute to the genetic basis of schizophrenia and thus is a part of the neurodevelopmental hypothesis of schizophrenia. Now let's discuss glutamate receptors. There are several types including the neuronal, IAT and the vesicular V-glut. Now there are at least 8 subtypes of metabotropic glutamate receptors organized into 3 separate groups. Now group 2 and group 3 receptors can occur presynaptically and thus function as autoreceptors to block glutamate release. And thus the drugs that stimulate these presynaptic autoreceptors acts as agonist and may therefore reduce glutamate release, and thus are potentially useful as anticonvulsants and mood stabilizers and may also protect against glutamate excitotoxicity. Now let's discuss group 1 metabotropic glutamate receptors. Now these are mainly located postsynaptically, where they hypothetically interact with other postsynaptic glutamate receptors, mainly to facilitate and strengthen responses mediated by ligand-gated ion channel receptors for glutamate during excitatory glutaminergic neurotransmission. Now the main receptors for glutamate are NMDA, AMPA and kainate. These are named after the agonists that selectively bind to them and are all members of the ligand-gated ion channel family of receptors. They tend to be postsynaptic and work together to modulate excitatory postsynaptic neurotransmission. Especially AMPA and kainate receptors may mediate fast excitatory neurotransmission, allowing sodium to enter the neuron to depolarize it. The NMDA receptors in the resting state are normally blocked by magnesium, which plugs the calcium channel. Now these NMDA receptors are interesting as they function as a coincident detector that can open to let calcium into neuron to trigger postsynaptic actions from glutamate neurotransmission only when three things occur at the same time. And these are: first, the glutamate occupying the binding site on NMDA receptor; second, glycine or D-serine occupying its site on NMDA receptor; and the third, depolarization occurring allowing magnesium plug to be removed. Interestingly, many important signals by NMDA receptors that are activated when NMDA calcium channel are opened include long-term potentiation and synaptic plasticity. Now let's discuss key glutamate pathways in the brain. There are about half a dozen specific glutaminergic pathways that are of particular relevance to psychopharmacology. And these are corticobrainstem, corticostriatal, hippocampal striatal, thalamocortical, corticothalamic, corticocortical and corticocortical, both direct and indirect. Now let's discuss these in some detail one by one. The first one is corticobrainstem glutamate pathway. It's a very important descending pathway and projects from cortical pyramidal neurons to brainstem neurotransmitter centers. And these include the raphe for serotonin, the ventral tegmental area, substantia nigra for dopamine, and locus ceruleus for norepinephrine. Now the direct innervation of monoamine neurons by these stimulate neurotransmitter release, whereas indirect innervation via GABA interneurons in the brainstem blocks neurotransmitter release. The second descending pathway is the corticostriatal one, and it extends from pyramidal neurons to striatal complex. Interestingly, it is known as the corticostriatal glutamate pathway when it projects to dorsal striatum, whereas it is known as corticoaccumbens glutamate pathway when it projects to specific area of ventral striatum, also known as nucleus accumbens. In either case, these descending glutamate pathway terminate on GABA neurons destined for a relay station in another part of the striatal complex called the globus pallidus. The third pathway is the hippocampal accumbens pathway, and as the name suggests, this projects from hippocampus to the nucleus accumbens. Interestingly, theories link this particular pathway to schizophrenia. These projections to the nucleus accumbens also terminate on GABA interneurons, that in turn projects to a relay station in globus pallidus. The next pathway is the thalamocortical pathway. This pathway brings information from the thalamus back into the cortex, often to process sensory information. The next pathway is the corticothalamic pathway. It projects directly back to the thalamus, where it may direct the manner in which the neurons react to sensory information. The next is the direct corticocortical glutamate pathway. It is a complex of many corticocortical glutamate pathways and are present within the cortex. Pyramidal neurons can excite each other within the cerebral cortex via direct synaptic input from their own neurotransmitter glutamate. And the last one is the indirect corticocortical glutamate pathway. In this, the pyramidal neurons can inhibit one another via indirect input, namely via interneurons that release GABA. Now let's discuss NMDA hyperfunction hypothesis of schizophrenia. A major current hypothesis for the cause of schizophrenia proposes that the glutamate activity at NMDA receptors is hyperfunctional due to abnormalities in the formation of glutaminergic NMDA synapses during neurodevelopment. This arises in part from observations that when NMDA receptors are made hyperfunctional by means of NMDA receptor antagonists such as phencyclidine and ketamine, it produces a psychotic condition in normal humans very similar to symptoms of schizophrenia. And genetic abnormalities can also make NMDA receptors and their synapses hyperfunctional. Now, what is so attractive about the NMDA receptor hypothesis is that, unlike amphetamines which activates only positive symptoms, PCP and ketamine also mimic cognitive, negative as well as affective symptoms such as social withdrawal and executive dysfunction. Another attractive aspect of this hypothesis is that it can also explain the dopamine hypothesis of schizophrenia, namely as a downstream consequence of hyperfunctioning of NMDA receptors. Now let's discuss the NMDA hyperfunction hypothesis in some detail. As just mentioned, this theory suggests that schizophrenia may be caused by neurodevelopmental abnormalities in the formation of glutamate synapses at a very specific site, namely at certain GABA interneurons in the cerebral cortex. Something appears to be wrong with the genetic programming of those particular GABA neurons that can be identified in prefrontal cortex as containing a calcium binding protein called the parvalbumin. These parvalbumin containing GABA neurons are faulty postsynaptic partners to incoming glutamate input from pyramidal neurons in prefrontal cortex and thus form defective NMDA receptor containing synaptic connections. Thus, to summarize, they have hyperfunctioning NMDA receptors on their dendrites. They have defective synapses between the glutamate neuronal axons and the GABA interneuronal dendrites, and thus faulty glutaminergic information coming in to the GABA interneurons. This so-called disconnectivity may be genetically programmed from a variety of faulty genes that all converge on the formation of these particular NMDA synapses. As a consequence of this disconnectivity, they also have deficits in the enzyme that make their own neurotransmitter GABA, causing a compensatory increase in the postsynaptic amount of alpha-2 subunit containing GABA receptors of the postsynaptic axon initial segment of the pyramidal neurons they innervate. When these parvalbumin containing GABA interneurons fail to function properly, they do not adequately inhibit key glutaminergic pyramidal neurons in the prefrontal cortex, causing those glutamate neurons to become hyperactive. Thus, one sick synapse in a neuronal circuit can affect the whole circuit, from GABA interneurons and the glutamate neuron it innervates to downstream dopamine neurons and beyond, as is shown in the flowchart. Linking the NMDA hyperfunction hypothesis to the dopamine hypothesis of schizophrenia, positive symptoms. The most relevant to schizophrenia are the glutamate pathways that regulate mesolimbic and mesocortical dopamine pathways. Now, the cortico-brainstem glutamate pathways regulate the output of glutamate from the cortex to the brainstem VTA for both these circuits. It appears that the cortico-brainstem glutamate neurons that innervate only the dopamine neurons projecting from VTA to the nucleus accumbens, that is the mesolimbic pathway, directly innervate those specific dopamine neurons and thus stimulate them. And as previously stated, the dopamine hyperactivity of these downstream mesolimbic dopamine neurons is associated with the positive symptoms of schizophrenia, but is actually caused hypothetically by the disconnectivity in upstream glutamate neurons, and namely defective and hyperfunctional neurodevelopmental glutamate innervation of parvalbumin containing GABA interneurons at NMDA receptor containing synapses. It is also possible that the disconnectivity of the upstream glutamate neurons in the hippocampus contributes to the downstream mesolimbic dopamine hyperactivity via 4 neuronal circuits. And these are as follows. The disconnected and defective hippocampal parvalbumin containing GABA interneurons going to the hippocampal glutamate neuron projecting to the nucleus accumbens, then that neuron projecting to 2 GABA spiny neurons in sequence. The first GABA spiny neuron going from nucleus accumbens to globus pallidus, and finally the second GABA spiny neuron going from globus pallidus to VTA. Now, loss of this adequate glutamate function at parvalbumin containing GABA interneurons in the hippocampus could lead to hyperactive glutamate output from glutamate neurons that project by this circuit to the mesolimbic dopamine neurons in the VTA, with the consequential dopamine hyperactivity and positive symptoms of schizophrenia. And stimulating 2 GABA interneurons in sequence has the net effect of disinhibition, that is, inhibition of inhibition at the VTA, the same result as direct stimulation. And understandably, the bottom line is that excessive upstream glutamate output from either the prefrontal cortex or the hippocampus may contribute to downstream dopamine hyperactivity and thus positive symptoms of schizophrenia. Now let's discuss linking the NMDA hyperfunction hypothesis to the negative symptoms of schizophrenia. It appears that different cortico-brainstem glutamate neurons regulate those unique dopamine neurons in the VTA that project only to the prefrontal cortex, that is the mesocortical dopamine pathway. The cortico-brainstem glutamate neurons destined to regulate mesocortical dopamine neurons in the VTA do not directly innervate them. Instead, the glutamate neurons do so by indirectly innervating an inhibitory GABA interneuron that itself innervates the mesocortical dopamine neurons. Thus, activation of these particular glutamate neurons leads first to activation of GABA interneurons, which then inhibits mesocortical dopamine neurons. And thus, dopamine hyperactivity of these mesocortical dopamine neurons is associated with negative and cognitive symptoms of schizophrenia. It is hypothetically caused by the same upstream disconnectivity of the glutamate with GABA interneurons that cause the hyperactivity of the mesolimbic dopamine neurons. Namely, the neurodevelopmental abnormality in the glutamate innervation of parvalbumin containing GABA interneurons at the NMDA synapses. Neurodevelopment and genetics in schizophrenia. Now, what seems a very obvious question now is what causes schizophrenia? Nature, that is genetics, or nurture, that is the environmental or epigenetics? The answer seems to be both. It seems more likely that schizophrenia is a conspiracy among many genes and many environmental stresses to cause abnormal development of brain connections throughout life. Not surprisingly, there is no single gene for any specific psychiatric symptoms, behaviors, personalities, or temperament. Today, mental illnesses are thought to be linked in part to someone inheriting an entire portfolio of many genes that carry risk of mental illnesses. Multiple risk genes, any one of which alone may be clinically silent until stress from environments puts a load on these defective genes. And these can be any kind of stress, traumatic experiences, learning experiences, sensory experiences, sleep deprivation, toxins, and even drugs. And these are all examples of how normal genes, such as those that regulate the information and removal of synapses, are turned on or off by environmental stresses. Cannabis seems to be particularly malicious environmental stressor to those vulnerable to schizophrenia. These are all examples of the notion of experience-dependent development of synaptic connections, something that is hypothetically abnormal in schizophrenia. Best evidence for the role of disconnectivity of genes in schizophrenia is multiple genes that regulate not only neuronal connectivity in general, but glutamate synapse formation and removal in particular. These genes include disbindin, neuroglin, ERBB4, and DISC1. Disbindin is also known as dystrobrevin binding protein 1 and is involved in the formation of synaptic structures and regulation of activity of the vesicular transporter for glutamate, that is VGLUT. Neuroglin, on the other hand, is involved in neuronal migration, genesis of glial cells, and subsequent myelination of neurons by glia. They also activates an ERBB4 signaling system that is co-localized with the NMDA receptors. Now, coming on to ERBB4 receptors themselves, they may be involved in mediating the neuroplasticity triggered by NMDA receptors and to provide both structural and functional regulatory elements for neurotransmission and for NMDA receptors. Both disbinding and neuroglin impact the formation and function of the postsynaptic density. And finally, DISC1 gene, that is disrupted in schizophrenia 1 gene that makes protein that is involved in neurogenesis, neuronal migration, dendritic organization, and also affects the transport of synaptic vesicles into presynaptic glutamate nerve terminals, and regulates cyclic AMP signaling, which affects the functions of glutamate neurotransmission mediated by metabotropic glutamate receptors. The bottom line is these all affect synapse formation. Also, NMDA receptor numbers by altering NMDA receptors trafficking to the postsynaptic membrane, NMDA receptor tethering within the membrane, and NMDA receptor endocytosis that cycles receptors out of the postsynaptic membrane to remove them. The other risk genes implicated are the genes for DAOA, that is D-amino acid oxidase activator, that codes for a protein that activates the enzyme DAO. As previously stated, DAO degrades the co-transmitter D-serine that acts at glutamate synapses and at NMDA receptor. This in turn would alter glutamate neurotransmission at NMDA receptors. Another gene is RSG4, that is regulator of G protein signaling. It impacts metabotropic glutamate receptor signaling through the G protein coupled signal transduction system. When glutamate synapses are active, their NMDA receptors trigger an electrical phenomena known as long-term potentiation. Now this LTP leads to structural and functional changes of the synapse that makes neurotransmission more efficient, sometimes called strengthening of the synapses. This includes increasing the number of AMPA receptors. These are important for mediating excitatory neurotransmission. This leads to robust neuroplastic influences. There's an old saying that goes like, nerves that fire together, wire together. If something is wrong with these genes, such a synapse would be weak, theoretically causing insufficient information processing in its circuits. This strengthening or weakening of a glutamate synapse is an example of activity-dependent or use-dependent or experience-dependent regulation of NMDA receptors and functionality at glutamate synapses. This may also explain why the course of schizophrenia is progressive and changes over time for most of the patients, namely from an asymptomatic period to a prodrome, to a first break psychosis with robust treatment responsiveness, to multiple psychotic episodes with declining treatment responsiveness, to a state of pervasive negative and cognitive symptoms without recovery. This continues throughout the life as a sort of ongoing remodeling in response to what experiences the individual has. Another important aspect is whether a given synapse is eliminated or maintained. If critical synapses are not adequately strengthened in schizophrenia, it could lead to their wrongful elimination, causing disconnectivity that disrupts information flow from circuits. Normally, almost half of the brain's synapses are eliminated in adolescence. Competitive elimination of weak but critical synapses during adolescence could even explain why schizophrenia has onset at this time. In adulthood, you may lose or replace about 7% of synapses in your cortex every week. As previously stated, abnormalities in the following genes may lead to lack of critical synapses being strengthened, that is, the dysbinding, the neuroglin, and the DISC1. And thus, critical synapses may be mistakenly eliminated during adolescence. Neuroimaging circuits in schizophrenia. fMRI can uncover abnormal information processing in brain areas linked to cognitive and emotion by putting a load on brain circuits and thereby perform a type of psychiatric stress test while visualizing the activity of the brain circuits. This can most readily detect the activity of neurons near the surface of the brain, which are mostly pyramidal neurons in the cortex. The activity of these neurons is the first leg of various brain circuits, especially the glutamate neurons acting within feedback loops from the cortex to the striatal complex, with that information relayed to the thalamus via GABA neuron, then back to the cortex again via another glutamate neuron as a cortico-striatothalamo-cortical or CSTC feedback loop. Brain circuits like these are information processing engines that are activated by various tasks or loads placed upon them, and seeing them light up may be literally watching the brain think. Functioning in the brain is topographical. For example, the DLPFC is thought to be most closely linked to cognitive functioning such as problem solving, whereas the VMPFC along with amygdala is thought to be most closely linked to emotional functioning such as mood. Patients with schizophrenia cannot adequately recruit the hippocampus during memory recall. It appears that the prefrontal cortical or hippocampal dysfunction in schizophrenia is more complicated than just hyperactivation or hypoactivation, but might be better characterized as out of tune. Patients with schizophrenia appear to utilize greater prefrontal resources when performing a cognitive task and yet achieve lower accuracy because they have cognitive impairment despite their best efforts. When performing poorly, schizophrenia patients do not appropriately engage and sustain the DLPFC, and this shows hypoactivation. To perform near normal, patients with schizophrenia engage the DLPFC but do so inefficiently, recruiting greater neuronal resources and thus hyperactivating the DLPFC. Thus, the DLPFC circuits in schizophrenia can be either underactive and hyperfunctional or overactive and inefficient. The unaffected siblings of patients with schizophrenia may have the very same inefficient information processing in the DLPFC and might have some mild degree of cognitive impairment, but not enough of the risk genes to have the full syndrome of schizophrenia itself. Schizophrenics has impairments in the ability to identify and accurately interpret emotions from overt sources, including facial expressions. This may be due to inefficient information processing within the VMPFC and amygdala and can be measured by imaging the response of the amygdala to emotional input, especially from facial expressions. Schizophrenics may not activate the amygdala in response to scary, fearful, or emotionally charged faces and thus have lack of emotional processing. It may represent as distortion of reality as well as impairment in recognizing negative emotions by schizophrenics. Failure to mount a normal emotional response to a scary face can also represent an inability to interpret social cues and lead to distortion in judgment. On the other hand, a neutral phase or a neutral stimuli may provoke a patient with schizophrenia. Activating emotional processing in amygdala when it is inappropriate may accompany the symptoms of paranoia and can lead to impaired interpersonal functioning, including problems in social communication. Imaging, genetics, and epistasis. It's interesting how single genes can alter the efficiency of information processing. Individuals with variation of gene COMT that has higher enzymatic activity called VAL has lower dopamine levels in DLPFC and thus less efficient information processing there, compared to individuals with a variation of COMT with lower enzymatic activity and thus higher dopamine levels called MET. Thus they say don't have tea with VAL. Now it's very interesting that the VAL variant of COMT by itself may or may not consistently alter DLPFC activity during a working memory test in schizophrenia compared to individuals with the MET variant of COMT. But when combined with another genetic variation of another enzyme that independently reduces the availability of dopamine in prefrontal cortex, there is a more robust decrease in dopamine in DLPFC, demonstrating the epistatic interactions. And that variant is the T form of the enzyme methylene tetrahydrofolate reductase or MTHFR, involved in the formation of dopamine and in regulating the activity of COMT at the level of gene expression. And you can remember this as you may want to have tea with Valerie, but you don't necessarily want to have tea with Val. Be careful of the alphabet soup. Another example of epistasis is how the efficiency of information processing in DLPFC gets worse if you combine 2 or 3 of several of the schizophrenia risk genes that have horrible sounding names from various letters of the alphabet. Some examples of these genes are NRG1 or neuroglin, ERBB4, AKT, etc. The NRG1 is involved in neuronal migration and synapse formation and can also impact the formation and function of the postsynaptic density. It also affects NMDA receptor numbers by altering NMDA receptor trafficking to the postsynaptic membrane, NMDA receptor tethering within the membrane, and NMDA receptor endocytosis that cycles the receptors out of the postsynaptic membrane to remove them. Now this NRG1 directly interacts with ERBB4 and activates the ERBB4 signaling system that is co-localized with NMDA receptors and interacts with the postsynaptic density of glutamate synapses and is involved in mediating the neuroplasticity triggered by NMDA receptors. And now finally coming on to AKT. It is a kinase enzyme and is part of the intracellular signal transduction cascade. It interacts with beta-arrestin 2 and GSK3 to regulate neuronal cell size and cell survival and to increase synaptic plasticity. And malfunctioning of AKT can lead to overactivation of GSK3 and lack of internalization of dopamine D2 receptors by endocytosis, contributing to dopamine hyperactivity.